Alright, so for this video, I want to go back over mitosis and meiosis just to really shore it up to make sure that you're feeling a little bit more confident and so that we can put meiosis in context of theoretical genetics. So we will be going over meiosis as well as mitosis. We will start looking at Punnett squares, which hopefully you guys have seen before. And then talk about different types of inheritance patterns. Today we'll look at 3 different ones, but we will have a few more to add on to that later in the week. So when we're looking at mitosis and meiosis, and I hope this is a good reminder for you, but I hope you already knew this. When we look at mitosis, we're thinking about from our head to our ptosis, right? It is responsible for your growth, for your development, and also what happens every time your cells need to heal or replenish cells that have maybe died, that have gotten old. So mitosis is a process that you guys are doing constantly, nonstop, all over your body, and it is what is making your body cells, the ones that are somatic, that is the 2N or diploid cell. And every time you do mitosis, the 2 daughter cells that are made at the end are going to be identical to what you start with, which is the parent cell. So you start with 2N and you end with 2N cells or 2 diploid cells. In the case of meiosis, it is going to be what is responsible for making our sex cells, so the gametes. It's going to be really important just in terms of creating genetic diversity, because each daughter cell that is made at the end of meiosis is completely different than any of the other 3 that are made, as well as different from the one that you started with, or the parent cell. And it's also going to produce offspring or daughter cells that have only half of the genetic information from what the parent cell had, so that when 2 gametes come together during the process of fertilization, what you have is back to your diploid number. And so then mitosis kicks in, that single cell grows and grows and grows and grows until it's time to start making sex cells. Then a little bit of meiosis, either just in the ovaries or just in the testes, and the circle of life continues. And so big picture here is when we're taking a look at mitosis or meiosis, they're both going to be starting out the same way, because we're thinking about the cell cycle and we're just looking at this part that is M. The rest of interphase is going to all be the same. So you have growth and replication of your DNA, or S phase for synthesis, and then some more growth. But in either case, mitosis or meiosis, you're going to start with a cell that is 2N or diploid, and so you start with homologous chromosomes. I'm going to show you what each pair of homologous chromosomes do, and in this case, in the diagrams that I'm showing you, we're going to look at it as if the cell is N equals 1, just one type of chromosome. But of course, you know that for humans, we are N equals 23. So all of the events that you see happening would be happening 23 times, like grander. So we start with our homologous chromosomes. That DNA is going to be replicated. And as a reminder here, we've got helicase that would come in, polymerase that would separate out the 2 strands and make 2 copies of the DNA. But remember that DNA is not some short little stretch of material. Instead, it's really huge, and it's so huge that it kind of starts to compact, right? We've got this coiling and then supercoiling, and each piece of DNA makes up just 1 chromatid. So by the time you are done with replication, you have made 2 chromatids, or sister chromatids, which are identical to each other. So sister chromatids have all the same genes and all the same alleles. Homologous chromosomes, shown in kind of the darker and the lighter orange color, are going to be same genes, for example, A, B, C, and D, but maybe different versions of each gene, or different versions, which we would then call alleles. So in mitosis, we start the process of mitosis again. So now we're kind of at the beginning of M phase. We've already done interphase, so this is not a brand new baby cell. It is a cell getting ready to make brand new baby cells. So at the beginning of M phase, we have sister chromatids that are joined together, but the homologous chromosomes are not joined together. They are going to be separate in the cell and each doing their own little thing. And then by the end of mitosis, that will have teased themselves out so that each new cell, each daughter cell that is produced, has exactly one of each version of each chromosome. So for example, we started in our original parent cell up here, right? This is what the cell would have in G1, so before synthesis has occurred. So we started up here with the parent with one dark and one light. We end with one dark and one light in each of the daughter cells. In meiosis, things are just a little bit different in that instead of starting with the sister chromatids together, but then the homologous chromosomes separate, we have everything lined up as tetrads. And those tetrads are all the same genes, but potentially different alleles between kind of the same color here. So for example, the 4 that are being shown might all have gene A, but in dark orange, maybe those are all big A, and in light orange, maybe they're all little a. So same genes, but different alleles, but then the parts that are the exact same color have all the exact same genetic information on them. So same genes, same alleles. And the reason that we have a little bit different stuff going on here is because of crossing over, or that process where some of the genetic material from the 2 alleles in the center of the tetrad have now switched over. So in meiosis, we're going to start out by separating the tetrad into 2 sets of mostly sister chromatids. Again, we're going to have just a little bit of area that are different, so they're not completely sister chromatids, but close enough, we'll consider them that for today. And that process right there is meiosis 1. Remember that mitosis has one nuclear division, right? All the chromosomes get sorted out one time. In meiosis, it's going to happen twice, so that by the time you're done with meiosis 2, you've now separated out the sister chromatids as well, and you've made 4 cells, each that only have half of the genetic information, and more importantly, that have different genetic information. So we started with a cell at the very beginning in G1, which was 2N, which was, right, diploid. That is your normal body cells. At the end of mitosis, we ended with exactly the same thing, 2N cells. So we started with, in humans, this would be 46 chromosomes, end with 46 chromosomes. But in the case of meiosis, we start with a diploid cell and end up with 4 unique haploid, or 1N cells. Okay. Just a little bit closer view on all the steps of mitosis. 4 in all. Again, we're starting with a 2N or diploid cell. We go through replication. That's going to make our sister chromatids. The sister chromatids are just kind of willy-nilly in the nucleus, and the nuclear membrane starts to break down, so really kind of all over the place in the cell itself. They get moved to the center or the middle of the cell during metaphase, and that is under the control of the spindle fibers. So here's the centrioles right here. Sorry, centromeres. Centrioles are on either side of the cell. These are going to make spindle fibers. And they do a little bit of a tug-of-war dance to get everything in the middle, but then they're going to tug hard enough that the centromeres that were holding the 2 sister chromatids together are pulled apart, A for apart. And finally, telophase occurs when 2 new nuclear membranes are formed around each pile of chromosomes here. Again, ending with exactly what you started with. So mitosis is going to start with diploid, end with diploid, boom. Meiosis starts the same way. You've got a diploid cell. It's undergone S phase. In this case, we're going to do it twice. So meiosis 1 is going to start in prophase. And unlike in prophase of mitosis, prophase 1 of meiosis has your chromosomes set up as tetrads. And again, we've had crossing over occur. And I've shown another set of chromosomes in the background just so you can compare prophase 1 to metaphase 1. So just like when we looked at mitosis, metaphase has everything lining up in the middle, but in this case, we've got it as a tetrad. So all 4 chromatids lined up together. That is the 2 sets of sister chromatids that are homologous to each other, that then get pulled apart during anaphase 1, and in telophase 1, 2 new nuclei are formed. So that by the time we start meiosis 2, we've already got 2 cells, and each of these daughter cells that is made at the end of meiosis 1 is going to be doing the same thing. In prophase 2, they're just getting ready to undergo the second part here. The nuclear membrane, if it formed at the end of telophase 1, is going to be disappearing again. The chromosomes are going to become visible and lining up and holding on to those spindle fibers. In metaphase 2, they get lined up in the middle of their respective cells. During anaphase, they're getting pulled apart. Anaphase 2, and then during telophase, that is the very last part of meiosis, is when they're going to have new nuclear membranes form, and that's going to produce 4 cells that are completely different than each other. Okay, so why does all of this matter? We need to understand what meiosis is doing with the chromosomes, and more specifically with genes throughout this process here. Sorry, I just want to make sure. There it is. Good, I'm still recording. Anyway, so by the time we have made the sperm or the eggs, everything's genetically different, and we need to make sure we're tracking what those genes are doing, in able to figure out like how genes get transmitted from one generation to the next. So we've got this process of fertilization, which is where 2 gametes come together. And why does this matter? Because how those gametes come together, first of all, explains how we can pass genetic information onto offspring, and this really should just say genes right here. And as well as like figuring out the combination of alleles that somebody has, those genotypes are going to be what are responsible for determining phenotypes. So we need to figure out how each phenotype is a result of each genotype. Okay, so the question that you're being asked for number 4 in your review says, if a parent has 2 different alleles for a gene, what % of their gametes will have each allele? So we're going to start with a parent cell that looks like this. I'm just giving you generic symbols to represent the different alleles because you've got to get used to seeing some different symbols, and we'll talk about that more later. But in this case, we've got one gene that has 2 different alleles. So this particular individual has one of each version, a heart gene and a puppy toes clubs version. So we've got hearts and clubs. And those are 2 different versions of the allele, and that's what the parent has. In your case, when you looked at your dragons, maybe it had big A, right? 2 of the same ones. Maybe it had little a, or maybe like this parent, it had 2 different ones. In either case, if this is the parent here, or the parent cell, and it has half heart and half club, then we need to see what's going to happen by the end of meiosis. So let's start out by just tracking the chromosomes as they go through meiosis, as if crossing over never occurs. We go to meiosis 1 through meiosis 2. And so we end up with 4 different cells, and essentially each one of these chromosomes just splits off to become 1, 2, 3, 4 different cells. So how about if we have some crossing over happening? We're going to see mostly the same thing, and by the end of them, we still have 2 heart alleles and 2 puppy toe alleles. How about if crossing over happens and it swaps out the genes that we're looking at too? By the end of meiosis, in this case, well, guess what? We still have 2 hearts and 2 clubs there. So regardless of what happens, if you start with one version of each allele, one of each allele, or that is one of 2 different versions, wow, I'm struggling, one of 2 different versions of the gene, then at the end of it, half of your gametes will contain the one gene and half will contain the other version of the gene, or the other allele. Okay? So why does this matter? And kind of going back, take a second and just talk to your partner. If a parent has 2 copies of the same allele for a gene, what percentage of their gametes will have each allele? So if we started this whole thing with, let's say, hearts and hearts, what would the outcome have been? And hopefully you can figure that one out. Okay, so let's say we have a mother and a father, and they are both this genotype, which is hearts clubs. That's their genotype. That's the genes that they have. I don't care what the phenotype is right now. We're just looking at the genotype. We're just looking at what is it that they possess, and then more importantly, what kind of gametes do they form? So a father who has hearts and clubs will have half of his sperm that contain the heart gene and half that contain the club gene or the club allele. Same thing we can say about the mother. Half of all of her eggs will contain the allele for the clubs, half will contain it for the hearts. And so if we're trying to figure out what all the possible combinations of offspring could be for this combination, or for these 2 parents, we have to take into consideration that half of the father's sperm will make one type versus the other type. Half of the mother's eggs will make one type versus the other type, if what she is starting with and if what he is starting with is one of each allele. So we could hypothetically have the sperm on the left, or one of the sperms that's carrying the heart allele, fertilize one of the eggs that's carrying the club allele, and that'll lead to an offspring, or a zygote, which just means the first cell after fertilization. So we could end up with a zygote that is heart club, or one of the sperm that carries the heart could fertilize one of the eggs that carries the heart allele, in which case the zygote would be heart heart. We could also have club club, or we could have club heart. So those are all the possible combinations, and you'd expect it in roughly the same ratio, because 1/2 of all of the dad's sperm carried the heart and 1/2 of all of his sperm carried the club's allele. Now you can draw all those arrows and drive yourself crazy, or more commonly what you're going to see is that we put it in kind of a graphical representation like this, and we really end up with exactly the same thing. We're just figuring out probabilities here. So we can use what are called Punnett squares, which all they do is determine the probability of offspring's genotypes. So if we know the parent genotypes, we can figure out what the kids are likely going to have based on all the possible combinations. And it doesn't matter if the male goes on the side or on the top, as long as you take the genotype and split up the 2 alleles so that they are the headers to the columns and the leaders for the rows. Okay, so if we start with clubs hearts, then clubs hearts here, we're just going to go ahead and take clubs and clubs, clubs and hearts, hearts and clubs, hearts and hearts. It's just like algebra. It's just like you've probably done this 1,000 times. And it doesn't matter which you put first, but if you're dealing with letters, typically they're going to either be in alphabetical order or capital letters first. Okay, so now that we understand how alleles get passed on to the offspring, we need to understand how they can interact to lead to particular particular phenotypes. And to understand this, there's a couple of vocab terms that we're going to introduce here. First of all, we're just going to be looking at genes that are on the autosomes for right now. And the reason is because the sex chromosomes get a little bit wonky, because males have one version of the X and one version of the Y. So it really changes up how inheritance can kind of work. So only the females have 2 copies of the same chromosome, that is 2 homologous or one homologous pair, whereas the autosomes, everyone has homologous pairs for all of them. So everyone is 2N, and therefore everyone has 2 copies of each gene that is on the autosomes. And so if those 2 alleles that they have are the same, for example, in this one, I told you I want to get a whole bunch of symbols in here, right? So instead of symbols, let's try color, right? So if you have red, then you have 2 versions of the same allele, and you are called homozygous for that particular gene. Homo meaning same, zygous referring to a gene. Same thing if you have white, you're still homozygous. So we could refer to these as homozygous red and homozygous white for the particular gene. In other words, just someone who has 2 copies of a particular gene, 2 of the same copy of a particular gene. If you are heterozygous, that means you have 2 different alleles for that gene. So for example, maybe you got red from dad and white from mom, or white from dad and red from mom, doesn't matter. In both cases, you're heterozygous, hetero meaning different. So you have 2 different alleles for the same gene. We're going to use those terms a lot in here, okay? So let's look at how genes can interact to make genotypes. The one that you're most familiar with, I'm sure, is an inheritance pattern that is called simple inheritance, or sometimes you'll hear me refer to it as Mendelian genetics, simply because of the fact that it is named after Mendel, Gregor Mendel, who is the founder of modern genetics. And you may also hear this just as like plain dominant and recessive, because of the fact that those are the traits that you have. So in simple genetics, the dominant phenotype, the one that is displayed from the dominant gene, is going to be displayed anytime that gene or that allele is present. In this case, we could assume that red is going to be the dominant one, because in individuals that are heterozygous, if you are red and white, those heterozygotes still express the red phenotype. That is, they still look red, they still have the trait that is associated with the dominant gene. And the only way to get the recessive phenotype is if you have 2 copies, or really if you're just lacking in the dominant allele. So the only way to become, to have a recessive phenotype is if you don't have the dominant gene or the dominant allele. Okay? So in this case, red would be dominant, and we would call someone who is red homozygous dominant. White is recessive, and we would call someone homozygous recessive if they had 2 of the white allele. And then the only time, and then you'd be heterozygous, if you have 1 of each allele. But not all traits are simple dominant and recessive. In fact, most aren't. Incomplete dominance refers to any time you have a phenotype that is in between. So when you think of incomplete, think in between. And that is, both alleles are going to be partially expressed. So if you have, for example, if you're homozygous red, if the red allele is partially expressed and the other red allele is partially expressed, you end up with a red phenotype. If you have 2 versions of the white allele, that is, you are homozygous white, then the white allele is partially expressed, so is the other white allele, and you end up with a white phenotype. What's different here than what we saw in simple inheritance is that heterozygotes are in between. So the red allele is going to be partially expressed, the white allele is going to be partially expressed, and the phenotype ends up being intermediate between either of the 2 homozygote individuals. So instead of red or white, you're in between. That is your pink. Codominant, co means both. So in the case of codominant traits, those occur anytime both traits are expressed. So if you only have red alleles, then you're only going to express red. If you only have white alleles, then you're only going to express white. But if you have both, if you're heterozygous, then you are going to not only express red, but you're also going to express white. And maybe this comes out as stripes or spots or any other thing. So there's like different flowers, for example, that kind of have different splotches of 2 or more colors, and that is because those alleles are codominant. So I talked to you a little bit earlier about using different symbols. Typically, with simple dominant and recessive, you're going to just give 1 letter, and you're going to have 2 versions of that letter to represent the dominant and recessive. The capital letter represents the dominant allele, and the lowercase letter represents the recessive allele. So in this case, we could say big A is red, and so someone who is homozygous dominant would be big A, big A. someone who is homozygous recessive, we assume that little a is white, and therefore they are little a. And then the heterozygous has one of each, and typically, again, we are going to put the capital letter first, okay? When we get into incomplete dominance or into codominant traits, things get a little bit weird. Typically, we're going to use two different letters, and I can't even say typically. Sometimes we're going to use two different letters. If you are doing any kind of inheritance traits that deal with codominant or incomplete dominant, make sure that you only use two different letters if you are keeping them capitalized. For example, R for red, W for white. We have an individual here who is homozygous, which must mean they're big R, big R. We have an individual here who's homozygous white, which must mean they're big W, big W. And the heterozygous would be big R, big W. Do not ever do something like this. That is like wrong. Each one of these different symbols, which is capital R, lowercase r, capital W, lowercase w. Oops, my bad. Those are all different symbols, and those would all represent different alleles. But in this case, it's only two different alleles, either red or white. Red or white. So do not use 4 different symbols to mean 2 different alleles. So that's just kind of my warning for you. Okay. Anyway, the goal for today, hopefully you've gotten up through number 12. You should do a quick check with me before you move on, but your goal for today is to start practicing some of these Punnett squares with your partner and finding out what is the likely genotype and phenotype ratio of your offspring. In other words, if you and your significant other in the dragon world were to have like 10,000 babies, roughly what proportion of them would you expect to have each of the given traits, given what you each have? So that's what you'll be practicing today, specifically with codominant, incomplete dominant, and then just simple Mendelian genetics. If you've got questions, let me know. Don't forget to hit subscribe and like and the little alarm button-y thing so that you can get notifications. And there it is. I'll catch you on the flip side. We are done.